Let me start by thanking all of you for coming. I know everybody's really busy at this point. There's a lot going on all over. I hope you're all safe. Hope your friends and family are all safe. But yeah, really, thanks for coming out. Really excited about this group that came together. I was really shocked with the interest when I put out the original tweet, and all the people that got back to me from all different fields, from archaeology like myself and paleoanthropology, but geochemists and biologists and botanists and paleontologists, and even more. It's really fantastic that there's so much interest in this line of research, and I know human evolution, everyone's interested in that. So really excited that all of you were able to come out and learn something new, hopefully. So thank you all. I hope that you do learn some new stuff today. I hope we can have a pretty good discussion later on. And then if you have questions, you can always ask me here or later on an email or whatever. And who knows, we'll go from here. The plan for today is to give you an overview of leaf wax lipid biomarkers and how they're applied in human evolution. I don't know all of you. I know some of you do this line of research, and some of you have done this in East Africa, which is fantastic. Some of you might just be starting out as undergrads or grad or PhD students, and you might want to know a little bit more. So for the goal today was to really introduce this line of research. If you have never heard of it before, if you've heard of it and you're really interested in adopting it into your own research agenda, hopefully this will add a little bit to that. And then we can go from here in further meetings or correspondence in other regards that way. So I hope that sounds good. If you do have questions as I'm talking, maybe just pop them into the group chat, so then I can see them at some point and respond to them, as opposed to me trying to stop when I see a hand waved or whatever. Does that sound all right? This is the first time I've ever done this, so just bear with me a bit. I'll give you a brief background on myself. I just got my PhD, well, about a year ago now, at the University of Calgary. I did my dissertation on early Acheulean environments at Olduvai Gorge in Tanzania. And since October, I've been working at the Max Planck Institute for Human Sciences here in Jena, in Germany, doing a few different projects in Lesotho, Kenya, Saudi Arabia, still working in Tanzania, but doing leaf wax stuff. Before that, before my PhD, I was working at a research lab in Rhode Island. I grew up in New England, and I have some of my colleagues here for that. And I've been doing the leaf wax stuff for almost 10 years now. Before I did Africa, I was working in China. So I've been doing this for quite a while now, and the time has been going by really too fast. So yeah, so unless there's any questions before we even get going, I'll just jump into the PowerPoint. Hopefully it'll take 25 minutes, a half hour, and then we can chat about a few things with all of us. Sound good? Okay. All right, so like I was saying, today what I really wanted to do was use this discussion, use this presentation as a jumping off point for this type of research, where we can look into collaborations down the line, or engaging discussions in platforms like this, or even set up mini workshops once we're able to meet again. But really, it was designed when I first came up with the idea to explore this line of research for archaeologists who may be interested in including a new type of paleoproxy into their research, but also incorporate the geosciences, geochemistry, people like that who are really pioneering this line of work, to then help us develop new research projects to target human evolution specific questions. So we're going to start today by really describing what are biomarkers, at least in the ones that I'm interested in. So what are they? So biomarkers is a catchy term for biological specific marker molecules. They're basically different compounds, chemical compounds composed of carbon and hydrogen and some other elements that indicate the past existence of life. We find them in sediments, paleosols, rocks. We can get them from fossils, and they're really just indicators of the existence of life at one point in the past, or even in environments today that might have been different 100, 200, 300 years ago. And they're really useful because they give us an idea on their biosynthetic origins, and then processes that have happened since they were deposited. So there are quite a few different biomarkers. The archaeologists, we're mostly familiar with things like carbohydrates if we're looking at starches, DNA and RNA, how we can sort of reconstruct our ancestral DNA going back last 100,000 years or so. Lipids, pottery lipids, animal fats, waxes, things like that. And then proteins. Proteomics is a big field right now in archaeology, and it's really starting to push the boundaries of how far back in time we can get. But there's some of these other biomarkers that really go back quite a bit in time. Some of these like chitin protein complexes, which go back almost 430 million years. When an animal and plant dies, as you can see our little diagram up here in the top, or even when they're alive, they leave a little bit of themselves behind. Trees shed their leaves and that get into the ground. And as they do this, they're leaving behind specific biomarkers. And then oftentimes they're buried, and they can be buried very quickly, and then they're preserved over long terms. Other times they might get stuck in the zone of bioturbation for quite a bit. They might get broken down by bacteria or fungi, and the original molecule might get rearranged. There might be some things happen to it chemically before then it gets preserved. And then, we come along, hundreds of thousands or millions of years later, and then we uncover them, these biomarkers, and we have to use these to study the past, use them as paleo proxies to understand environmental changes that have happened in the past. And for us in the archaeological sciences, we use these to answer questions related to human evolution or human populations through time. Now, a few issues with some biomarkers. We have to taphonomically normalize them. Things happen through their isotopes or their chemistry over time. They actually get broken down a long time by diatogenesis, by heat and pressure, and all we might be left with is a fragment of that original biomolecule. So what we then have to do is use that fragment to reconstruct the past, and that can be quite challenging. Fortunately, for the work that we're going to be discussing today, really we're only going back 2 or 3 million years, so we don't have so much to worry about in terms of diagenesis, in terms of this breaking down by heat and pressure. But we will briefly talk about biodegradation, because this can be seen really rapidly with some of the molecules we'll talk about today. Okay, so most people, I would think, are familiar with lipids, of the term lipids. These are just hydrocarbons that are insoluble in water. They have, for the most part, a low chemical reactivity, which allows them to last so long. And they're ubiquitous. They're in plants and animals. They're all throughout the environment. They survive really long as paleoproxies. On the last slide, let me just go back one. You can see here where I have the N-alkanes listed. There have been some evidence finding N-alkanes going back 55 million years, the early Eocene, and that's really, really amazing, really cool. But those instances, you really need rapid burial or just perfect burial conditions. But really, some of these lipids that we'll talk about, the biomarkers that we're interested in today, really can survive long term. So we're interested in the waxes, leaf wax lipids, and the two biomarkers that have been used mostly in this type of research are the N-alkanes and the N-alkanolic acids, and to a lesser extent, the alkanols. But there are a few other, actually quite a bit of other chemical compounds within the waxes of plants that we can get, really interesting paleo-environmental information out of. So when I talk about the waxes, when I talk about these biomarkers, what we're really focused on is this epicuticular wax deposit on the outer surface of the leaf. So if you look here on this dumb cane, and you can see on the outer surface of the leaf, it's really shiny. That's because it has a really waxy outer layer to act, we'll get to that in a second. And then if you look at the micrograph, the scanning electron micrograph, you can actually see the waxy deposit on the outer surface. So this is it. This is what we're really interested in when we talk about leaf wax biomarkers, the compounds within these waxes. So when a plant dies or it loses its leaves, these waxes go into the ground. They either get buried on site and then get added to the soil record or the sedimentological record. While at the same time, they can get blown long distances by wind and water and transported into lakes or oceans. And then we can use them to reconstruct large-scale environments. Now, when plants are growing, they use these waxes on the outer surface to protect against excessive UV radiation, just like our skin, insect and fungal attack, water loss in dry periods or when it gets a bit hotter, and then wind abrasion, anytime the wind might be blowing sand or any other type of particulates. The waxes really protect the leaf from excessive damage, so then the waxes can then grow and the plants can then grow. Within those waxes, we have these different saturated biomarkers, the three that I mentioned a few minutes ago, the alkanes, the alkanols, and the alkanoic acids. You can see that there's straight chain hydrocarbons, meaning that you just have a carbon bonded to carbon with hydrogens, the maximum number of hydrogens bonded to each carbon. So the ones that have been given the most attention through time are the alkanes. They're probably the easiest to work with, as we'll see in a bit. And they last really long term. But some of the other ones, the alkanols with the hydroxyl group at the end, or the N-alkanoic acid with the carboxylic functional group, they also last quite a bit, and in certain situations they actually preserve a lot better than the alkanes. So it's really fortunate that we have these different biomarkers we can work with in case something happens to one of them. It's biodegraded, it's just not in our samples. We have these different options that we can go with. They have strong covalent bondings between the carbons and the hydrogens, and that's actually one of the reasons why they survive so well long term. It's really difficult to break them apart without really elevated temperatures, 100 to 150 degrees, increasing pressure, or specific enzymes secreted by microbial activity to break them down. Now, for like many of our proxies, biomarkers can be found throughout the environment. If we're interested in human-related questions or human evolution-related questions, we want to look specifically at archaeological sites. And that's great because these are going to be deposited wherever plants are growing. And that could be right around where our specific archaeological sites are. But like I said before, they could be spread long distances by wind and water. They might get into lake environments or ocean environments. And then what's great about that is we can compare our records. We might have our on-site soil chemistry and micromorphology that we can use to then compare to lake cores or ocean cores. So then we might have a very localized environmental or climatic signal compared with a more regional and then maybe even a global signal or continental scale signal. Okay, so that's just sort of our introduction to biomarkers. We can talk about those more later, but just for now, I think the major thing we want to know is that our two main plant leaf wax biomarkers that we're going to be focused on are the N-alkanes and the alkanoic acids. There are other biomarkers we can use, and they have other functions, but today I just wanted to keep it a little bit more simple and just focus on those. Now, as a biomarker, we can use the alkanes as is to reconstruct past environments, looking at their abundance or their carbon chain length, how many carbons there are, to get an idea of past environments. But lately, and by lately I mean within like the last 20, 25 years, there have been some great advances in technology, precision in instruments for measuring these, and we've really shifted towards using the isotopes of these compounds to reconstruct past environments. Because we know from modern plants or modern environments how vegetation, how trees, how grasses, how bushes, how they incorporate local signals into their structure. as they're synthesizing these waxes to create this protective barrier as they're photosynthesizing. So they incorporate, local atmospheric conditions, local water conditions, things that are related to temperature and aridity. And we can study that real time with living plants or greenhouse experiments and from different environments. And then we can use that to reconstruct things in the past. So when we do recover these biomarkers in 2 million years old sediments, we can get an idea of what was going on climatically during the emergence of the Acheulean, for example. So there are a number of different things that actually impact the carbon and hydrogen. Oh, yeah, I should probably have mentioned that in this slide. So here, if you look here, for those of you who might not be familiar with isotope analysis, what we're looking at here is the ratio between heavier carbon, carbon-13, and the normal carbon, carbon-12. And we write that ratio out using the delta signal. And delta D is the ratio between heavy hydrogen and regular hydrogen. So really what we're looking at are changes in these ratios, heavy to lighter isotopes, and how that can be used to reconstruct past climates and environments. And we'll take a look at that in a minute with some of these factors. So there are quite a few factors influencing the isotope values in the plants. For carbon, the main ones are really the atmospheric CO2. What is the isotopic value of atmospheric CO2, plus the specific photosynthetic pathway, C3 plants versus C4 plants. For delta D, for hydrogen, really we're taking a look at source water, where that water is coming from. Is it from ocean evaporation? And then how far is it being transported inland, and is it losing out on its different isotopes? How the isotope values are changing with each successive rainfall event. There are a lot of other factors going on. We'll touch upon a few of them, but temperature, sunlight, aridity, things like that can change the concentrations in the plants, both for water and carbon. Altitude, latitude as well. So we'll take a look at some of these. If you have some questions, remember, keep them in mind, and we'll go through them later on. But for carbon, like I was saying, the atmospheric carbon really has a lot to do with the values in plants and living plants and plants in the past. So today, our atmosphere has 415, maybe a little bit higher, a concentration of carbon. And the 13C value is about -8 or so. And that's because we're burning fossil fuels and we're putting these negative CO2 back into the atmosphere from these plants that were growing 300 to 400 million years ago. So we're actually lowering or depleting the concentration of our atmosphere. Now, C3 and C4 plants, when they photosynthesize, they take in carbon from the atmosphere and they fractionate it. So C3 plants have a much larger fractionation factor. It's -C21. That is because they discriminate against the heavier carbon-13 during photosynthesis. C4 tropical grasses, on the other hand, although they still discriminate, it's not as intense, and it's really only about -6 per mil. So this symbol here, we just call it per mil. So because of that, we have these different fractionation factors. And then when we look at bulk carbon values for plants, C3 plants have a bulk signature of about -28.1, while C4 grasses about -13 or so. So when we're looking at plants, that's great because then we can really compare the signature from C3 and C4 plants. So C3 here in green, C4 in blue. And this is one of the best things in terms of using the alkanes for biomarkers, that in tropical environments or really hot and dry environments with C4 plants grow, we could get an isotopic signal distinguishing between the two. C3 plants typically range between -27 and -40, but there are some instances that go up to -45. Where C4 typically between -14 and -25, -26 or so, which is really great. Yeah, there's a little bit of overlap, but for the most part, we can do really well in distinguishing these in paleo records. Now I've used the C29 and the C31 alkane because these are the most common compounds used in paleo reconstructions, and they're typically the most abundant in plants in both C3 and C4 plants. So just carbons, 29 carbons are part of the chain as opposed to 31. Yeah, so this is great. I mean, we can use carbon to really distinguish between C3 and C4 vegetation. And then in tropical soils, you know, East African acacia commiphora biomes, for example, because of soil dynamics and things we know about modern environments, we can actually use this to reconstruct an environment in the past. Now, soils, they'll be enriched, they'll have more positive values for the most part than the plants growing on them. And this has to do with a few different factors, but generally there's a +4 to +6 enrichment between plants growing on the surface and then the soils themselves. And this has to do with, rate of burial, bacterial activity, loss of plant matter, things like that. So we can actually use this for our reconstructions in the past to get a general idea as to what was going on through like soil chemistry and things like that. As for hydrogen, on the other hand, source water delta D, so from the oceans, the original value is about 0 or so. That's where our original moisture source is coming from. And as ocean water evaporates and moves inland, it typically loses more of the heavy isotope in rainfall, during the rainfall. So our initial rainfall events might be slightly depleted after losing out some of the heavier hydrogen. And with each successive precipitation event, it becomes more and more depleted. In the tropics, we call this the amount effect. As we have heavy rainfall, you get more and more depleted rainfall with higher precipitation. So when we're looking at our values in the past, anytime we have more negative or depleted isotope values, delta D isotope values, it might indicate more rainfall or a wetter period in the past. Temperature, latitude, and altitude, they all influence it as well. If we're thinking about areas in the northern hemisphere that might not have C4 plants, these factors themselves actually can cause depletion in the hydrogen isotope values because it takes more energy to evaporate heavier hydrogen. So in colder environments, we actually get depleted values with changes in temperature. So colder environments, glacial periods, for example, might have depleted hydrogen isotope values. Something else that we really must consider is the evaporation and evapotranspiration from soil and leaves themselves, because that actually causes quite a bit of enrichment in the plant leaves themselves. And it sort of goes hand in hand with rainfall, successful rainfall and evaporation events. And then finally, the degree of continentality, basically the distance from the source. As you move away from the ocean or away from large lakes, you sort of lose the heavier isotope with successive rainfall events. So as you go in. And then there are just a few secondary factors that also play quite a big role, and we won't go too heavily into them. I think the main takeaway is looking at those primary factors, the photosynthetic pathway and source water. But we do have some secondary factors like the taxonomy and phylogeny of plants themselves. Are they broadleaf or do they have needles? How does that play into a role in wax production or evaporation? How well are plants at retaining water during dry periods when they also can't shut down photosynthesizing and suffocate themselves? Sunlight intensity and duration, especially as we go into the summer months, how does this factor into causing water loss or stress on the plants? And then of course temperature. Another interesting thing in forests, whether boreal forest or deciduous temperate forest, canopy height also seems to play a factor with leaves closer to the top of the canopy being a little bit more enriched. And this probably has a lot to do with sunlight intensity and evaporation. Now for hydrogen, plant ecological life form, is it a tree, is it a grass, is it a bush, actually has quite a bit of, plays quite a bit of a factor, because these different types of plants have different root depths and they take in different water sources. And things closer to the surface might have more enriched values if things are evaporating quicker, as opposed to things that are getting groundwater from deeper underneath, which might not have any evaporative enrichment. So just keep these in mind when we talk about a few other things. But yeah, there are quite a few factors impacting our isotope values. And it can get quite a bit confusing, as opposed to just, oh, this is great, C3 versus C4. No, we actually have quite a bit of different factors influencing these values. And it can get complicated trying to reconstruct past environments when we know there weren't C4 plants, or we don't expect C4 plants for that matter. Fortunately, we do have modern studies, correlates, and referentials, like I was saying, greenhouses or living plants today, to really get at some of these factors and see how they influence the plants, and then we can use that for our interpretations of the past. Okay, so we have an intro to our biomarkers, an intro into the factors influencing their isotopes. Let's take a quick look at how we extract these biomarkers from sediments or paleosols or whatever, and then use them to tell us a story in the past. So just a general workflow of what it is. Sample collection, obviously, a lot of us go to the field, or we have samples shipped to us from field sites or coring, and then we have to work with those. We have to dry and homogenize them. We have to extract the lipids, purify those lipids into individual fractions, into individual biomarkers, and then analyze them with gas chromatography and mass spectrometry, or isotope ratio mass spectrometry. So a few different ways of collecting samples. We might have terrestrial outcrops, geologically defined outcrops that we can go and sample through different stratigraphic horizons. We might have lake and ocean cores that are picking up a more regional signal that we can then use to compare to our archaeological sites. And then we might have actual trenches, archaeological trenches that we can correlate with the artifacts themselves or fossilized material because of well-defined, geologic strata, for example. Depending on the sediments themselves, you might have a sandy or gravelly sediment, and the integrity of the outcrop might not be great, and you can only do 2 centimeter high samples with 4 centimeter gaps. Whereas in some silty or really fine-grained mud samples, you can take a sample every centimeter, for example. And then with ocean and lake cores, because of the drilling, maybe even sub-centimeter sampling, and that's really fantastic. And the greatest thing about our different methods is that we can take our trenches from on-site, from archaeology itself, and then compare with more regional and continental scale records to get a robust picture of localized climate or localized environment in response to global changes. Now, to extract the lipids, there are a few different methods. The kind of tried and true classic method that has been in use for, I don't know, 50, 100 years, if not more, is Soxhlet apparatus. It's a great technique for, you put your sample inside, you turn on the hot plate, you evaporate some solvent, the solvent drips down, collects, and then siphons back in, returning your lipids to the main flask. Now, the problem with this is, yeah, it's time consuming. It takes up to, I don't know, 72 hours for successful extractions, depending on the age of the material. It uses a lot of solvent, but it's fairly relatively cheap. But if you only have 3 or 4 or a few of them at a time, it can really take a long time to extract sample sets. Ultrasonic agitation, basically using an ultrasonic cleaner to use cavitation bubbles to release the lipids from the sediments. This, for labs just starting out, or if you're just thinking about getting into this, this might be your best bet to start out with. Relatively cheap for an ultrasonic cleaner that has like a 2-liter tank. You can get one for, you know, $500–600 American US dollars, which is about the cost of one SOx slit apparatus. So these are all relatively cheap. The problem with ultrasonic agitation is very hands-on and time-consuming. You have to be there for a number of hours, you know, transferring solvents and adding new solvents and things like that. And then lately, again, I think the first application was 1996 or so, but lately we've had more use with these pressurized or accelerated solvent extractions. These ones that are all automated. They use a little bit of solvent compared to the other techniques, and can really extract samples within, you know, an hour or two, and then really up to, I don't know, maybe 18 to 24 samples in a day. And that's really fantastic. It's hands off, and it does all the work for you. The problem is they are really expensive, and purchasing one to start really can set, you know, new labs back quite a bit. Oh, yep, oh, I see some people chatting. That's great. I love that you're actually responding to each other. That's perfect. And it's a great question Rachel had. What volume of sediment is required per sample? And as Kate, I should point out that Kate Wachowski, Dr. Wachowski, was actually the one who originally trained me in doing this type of work. So it's awesome that she's here and participating. For some of the samples that I worked with that are, you know, 1.7 or so million years old and aren't very lipid heavy, I was using up to, you know, 30–35 grams per SOx slit extraction. If you're using, you know, modern topsoils, you can really get away with, like here, you know, maybe 5 to 10 grams. And then some other stuff, really, it does. It depends on the organic content, and you can do. How do you know that ahead of time? Well, you can always test a few samples at different sample amounts to get a general idea of how much your sample, how much organic content your samples actually have, because it can really vary anywhere from, like you guys are saying here, 5 grams up to 35 or not more. Okay, can I close this? So thanks for having that chatting and answering those questions. That's awesome. Okay, so once we get our lipids and we have our total lipids, so our output, we use, sorry, let me just go back one slide if I can. The solvents, typically we use in a 9:1 ratio, or 90% to 10%, dichloromethane and methanol. Now these are organic solvents. And when we're doing these extractions, basically what we're doing is using the like-dissolved-like principle. So we're looking at polarity. So our lipids, looking at the polarity of our lipids compared to our polarity of our solvents. And DCM has a polarity of about 3.1, and methanol is about 5. And when we mix them in this ratio, it's actually perfect for extracting our leaf waxes from sediments. So yeah, you typically use 9:1 in each of the different techniques. And then once we get the extraction, you can see this sort of our little diagram here. Once we have the extraction, we evaporate that down with a vacuum evaporator or nitrogen gas. And then we have just our lipid residue, our total lipid extraction. And then from there, we take that lipid and we subject it to a few different column chromatography techniques. Now I use a subset. I always start with aminopropyl columns. This is, they have a nitrogen-based compound within the silica gel itself. It's a functional group. And really it's perfect for separating out our neutral compounds, which contain our N-alkanes, our acid, and then our more polar, heavier type lipids. Because the aminopropyl columns themselves retain acids until you pass over basically a weak acid, acetic acid, for example, over the column, and then it releases the alkanoic acids, for example. So I always start with aminopropyl columns, separate out into 3 fractions, and then I typically work with the 2 the neutral and the acids fractions. Now, the one thing to note, I won't go into it too much today, but you do have to methylate the acids to make them so you can actually read them on the GC, because that functional group at the end, the carboxylic acid, can bind to the column, the GC column, and get stuck a bit. So we have to kind of change that, change their chemistry quite a bit. Anyway, after that, we do our more typical silica gel column chromatography, and this has been in place for what, 55 years now, if not a little bit more, and used to extract lipids. And basically what it entails is using silica gel, a fine mesh silica gel, and you basically construct, use a 5-inch pipette, and you just fill it with about 1 gram or so of silica gel, and then you pass your samples over it. And based on the polarity of the solvents that you're using, either your sample passes through the column. If we're looking for our N-alkanes, we could just use hexane and it passes through. Or it binds some of the more polar compounds and then they don't get passed through until you use something like methanol. When I do my columns, I typically like to combine silver nitrate infused silica gel. So I'll take 10 grams of silica gel and infuse it with about 30 milliliters of silver nitrate solution. And then I'll build my columns with the silver nitrate at the bottom and then straight normal silica gel on top. Now, the whole point of this is when you do have some degradation in your compounds, or if you do have compounds that have multiple bonds, so they're not straight chain hydrocarbons, they can cause interference when doing GC gas chromatography analysis. So the silver nitrate is really good at bonding with these double bonding carbon compounds, and it basically cleans up your profile, your chemical profile a bit. Some people might use 2 separate columns. They might do a silica gel column first and then a silver nitrate column afterwards. I typically just do them all in one, and usually, get some pretty good results of cleaning up some unwanted compounds. And you can see over here, sort of an early stage separation where you put in your lipids. With, these ones have this sort of yellow color, and as they elute through the column, they sort of bleed out. The darker color stays at the top, and you can just sort of make out the difference between the straight silica gel and the darker silver nitrate here. And what we're doing here is then we're separating our different biomarkers into individual fractions. And then from here on out, we can evaporate these down as well and do some GC analysis. So the biomarkers that we're interested in, I was saying before, the alkanes and the N-alkanoic acids. So here the N-alkanoic acids is fatty acid methyl esters, and then here are alkanes up at top. And typically our alkanes have this nice odd over even pattern where we have our odd compounds, 29, 31, 33, dominating the even compounds. And then it's the opposite for the acids. You basically have the even numbered compounds dominating the odds. And that's great. So in terms of biomarkers, we can get a general idea of terrestrial plants having these higher chains, C27 to C35, or C28 to C34 compounds, and that's great just as sort of a qualitative assessment of past environments. And with some of our lower chains, more typical of aquatic plants or algae, or maybe some bryophytes or things like that. Then we can test the isotope values themselves. Looking at the fatty acids again, we can analyze the isotopes. And that would give us an indication, in this case, we're looking at something that is in the -23. And if you go back to those slides earlier, if we looked at C4 plants, they were between -14 and -26 or so. So this falls sort of in that middle range for C4 plants. And we can, kind of get an idea that that was probably a wooded grassland, for example, with a C4 dominance of plants. All right, let me just look at, before we maybe ask a few more questions or discuss a few things, but just a few case studies of some of the work I've done looking at biomarkers in human evolution. But we have a few people here that are really attending the talk. that are also doing fantastic work in East Africa, mainly in the Turkana Basin in Kenya and at Oldupai Gorge in Tanzania. And they're really doing some awesome work looking at changes in hydrology and how changes in Earth's orbital patterns around the sun or its axial tilt actually influence global rainfall patterns, and what that means for lake regressions or lake expansions or rainfall patterns in East Africa, and how humans responded to changes in their plant landscape. We've also had people looking at meter scales, 5, 10, 100 meter scales, changes in the landscape over these sorry, I'm reading comments. Changing these landscapes and what that means for human activity in a lake environment, for example. So the Turkana Basin are really famous for some of our earliest evidence of the Acheulean, if not our earliest evidence of the Acheulean stone tool industry at about 1.75 million years ago. Really, it's the biggest transition we see going from the early Oldowan to then the later Acheulean, will last for about 1 million years. It has an incredible human fossil record. Homo erectus, some of our earliest evidence of Homo erectus shows up here. And like I was saying, we have some really great work being done there using leaf wax biomarkers. Oldupai Gorge, on the other hand, really famous because of the leakies, and who have been, who had worked there for 40 years or whatever, and really research in the gorge has been going on for over 100 years since the first, Germans were there looking into the geology. And it's really famous. This is where Paranthropus boisei was first discovered and sort of changed the focus of human evolution studies to East Africa from Asia. And like I was saying, we've had some really cool work being done looking at ecosystem variability and what that means for human foods and how they change on the landscape. So really I want to just take a look at one quick case study that I looked at just to sort of bring it all together now, where I looked at carbon isotopes in response to lithics and lithic modified bones or human modified bones, and how we look at changes through time, plant landscape through time, and human activity through time. So if you follow this curve, basically it's just a C29 and C31 N-alkane isotope curve over about a 34,000 year period. And you can see down at the bottom we're a lot, minus 30 per mil, so much more C3. And at the same time you have abundant lithics in this period. And as you sort of go through time, you can see it starts becoming more positive. You get more C4 grasses, the landscape opens up, and as the landscape opens up, you actually lose much of the lithic material and human modified bones. So if we sort of go back to our little diagram we had earlier on and compare the our forest to a grassland environment to our lithics, we can see that as the landscape opened up, this area wasn't as attractive in terms of maybe resource availability or water availability or just protection from carnivores, for example. And as the landscape opened up and became a bit more drier, a bit more grassier, human activity reduced and was moved elsewhere, somewhere else probably with more grasses. And then just a little bit earlier in time, an Oldowan site. This is about 1.8 million years old at the FOK North site, right in the center of the gorge. And we can compare a few different sites looking at their hydrogen values. And we can see that here we had a really dense closed woodland that was probably getting its water sourced from streams or groundwater coming off for the highlands. There's a volcano chain just south of Oldupai, as opposed to another site that's sort of that classic picture of Oldupai that you also always see is called the Castle. Maybe 200-300 meters away from FOK North, and the hydrogen values there were actually quite a bit different. This is also a bit different in time, but when we look at these different values, you can really see quite a bit difference in hydrogen values, which may have something to do with rainfall intensity changes over time or sourcing of groundwater. And we have a lot of archaeology at FLK North, but at the castle itself, really, there isn't any archaeology. Okay, and then just to close up and just to get us discussing some things in our, sorry, I talked a lot longer than I was expecting. What biomarkers are really doing for human evolution studies is we can reframe old evolutionary questions. How humans responded to changes in their environment, local, at a local scale and a regional scale, and then compare that with, the archaeological record itself. We can take a look at things like our broadly defined habitat-specific hypotheses, something like the savanna hypothesis indicated in the background, and how that's been sort of refuted by some of these biomarker studies by showing these broad changes between closed and open habitats and humans adapting to these different environments. We can also look at the diversity of hominin habitats and food and how changes in the biomarker record correlate with changes in temperature from enamel isotopes or teeth isotopes. Again, some really cool work in Kenya looking at how, although human habitats might have diversified, human foods have always been diverse, so it might not correlate directly with the habitats themselves, or maybe it does, and it just says that humans have been always very diverse with their foods. And then we can look at climatically driven fluctuations in resource availability and what that means for human migrations or movements out of Africa into Eurasia. So these are just some broad things that maybe we can get talking about in our time left. But yeah, okay. Thank you for listening for that. Let me just look through does anyone have any questions on some of that? I know I went through pretty quick, but if there are specific questions you have, let me know. We can talk about them. Oh, so I see one question. Sorry, if you know what, I think it might be best if you have a question, just put it into the chat, and then I can read them. So I'm just going to go through the last one. One thing I didn't mention is that, yeah, leaf waxes are great on their own, but really I think it's best when you compare them with other indicators for past vegetation. Phytoliths, for example, the pollen record, because some plants, they produce a lot more waxes than others, and you might pick up that signal even though they're not really representative of the majority of the environment. So it's always great to compare your isotope record with other plant records to really get a more robust picture of the environment. So something I didn't say in our study of the early Etrulian, we compared the phytoliths to the isotopes as well as diatoms to really get a general record. And then for something like this, for the question pollen, for pollen, yeah, you might have a different signal. Pollen really does travel quite a bit by wind and water, and some species create a lot more pollen than others, and I think that really is very similar with biomarkers. Yeah, so I think, again, really it should be a comparison thing. Looking at another question here, what about the extraction from volcanic tuffs? That's pretty tough. No, sorry, bad joke. The thing with the tuffs is that as they're being put down, it's difficult for plants to really get a foothold in the chemistry of the tuffs. You might need a bit more soil formation. I know some people have done other isotope stuff, not so much the leaf waxes. Maybe people that have. I know there are some pioneering species, some types of ferns that really grow in these environments quickly, and there are specific fern biomarkers that can be used, and then maybe from there go back to the alkanes. So that is an interesting question, because in places like Olduvai, we do have a lot of volcanic tuffs, so it would be interesting to see pioneering species after one of these volcanic events. Preservation in dry environments, that's interesting. I've seen some reports saying that because it's dry, there isn't as much bacterial activity, and you don't get as much breakdown in the zones of bioturbation right away, and you get a little bit more chance of burial. But what I've seen in Tanzania, in some of these dry environments, really there's sort of a weird interaction between soil chemistry, temperature, when it does rain, and microbial activity, all working to actually kind of biodegrade the biomarkers in what we would call topsoil, so they'd be relatively modern within the last 50 years or so. But I do know in places like Arabia, we're actually taking a look at some of these to see the preservation in some paleo lake cores. Yeah, again, it would have to do with the plants. If plants can really establish a foothold in these dry environments, and then long-term preservation. Are the sediments being carried by wind over long distances, or are they being buried fairly quickly? Yeah, so some great other questions. I see one about stalagmites. So these are other offsite proxies looking at the isotopes of, basically oxygen. We can use stalagmites and stalactites for precipitation records. And what's really great is we can use those because they can be well dated and they do record great hydrological signals. We can actually use those to compare with leaf waxes from on site. So you can compare the hydrogen with the oxygen isotopes from leaf wax, from stalagmites and stalactites. So I think that's one of those interesting concepts, is if you want to look at both on-site and off-site proxies, if you have access to do both alkanes and speleothems, definitely try and do it to see how hydrological conditions might change at a regional scale. Yeah, so another great question. I always do GCMS first. You sort of have to before you do the isotope ratio. You can do gas chromatography mass spec or gas chromatography flame ionization detection. These are just two different methods that we use. One, we want to know our concentration of our samples, of our biomarkers in the samples, because that'll inform us how much we need to inject or how much we have to concentrate our samples for the isotope machines. Up until recently, the IRMSs weren't as sensitive as a regular GCMS, so we always had to worry about concentrating our samples down. So really, to get an idea of the preservation and just the general quantities, always run your samples on GCMS first, and that'll give you an indication of biodegradation as well, and whether or not you need to do other type of cleaning up processes to get rid of some of these degraded compounds. So there are a few different types of microbes that are especially detrimental. Alex Brittingham, who is here, maybe can talk about this. He did a study looking at how leaf waxes can break down within a month's time in a laboratory setting. I know in East Africa we have some filamentous bacteria that grow near the rift lakes that are especially rough in breaking down some of these bacteria. But I don't know if Alex, Alex, you hear me? Would you want to comment on that?
Yeah, I can talk about that briefly. Yeah, so we did a study in our lab where we stored samples that we had collected for 3 months, I think it was, and found that over time, the samples that we had stored frozen, we found changes in the auto-reven predominance, the average chain length, and the hydrogen and carbon isotopes over time. And then we measured the bacterial DNA that were present, and we found that bacteria that had enzymes specifically for alkane degradation had proliferated in the samples during their storage. And there were a few other studies back in the 80s that found similar results, just looking at the average chain length and auto-reven predominance changes, but without the isotope results. So yeah, there's kind of a growing literature that bacteria can actually affect the isotopes and distribution of chain lengths in the alkanes.
Yeah, I think this goes back to what we were saying about taxonomic normalization, because it's really easy to just say that, oh yeah, these biomarkers last long-term in sediments and we go and collect them, and what the isotope signal says is what's going on in the past. But like Alex was saying, there have been a few recent studies looking at biodegradation and soil profiles, just going down 40, 50 centimeters and looking at the changes in isotope values based on degradation from bacteria. And yeah, they do have these specific enzymes for breaking down hydrocarbons and alkanes, for example, and it changes it. So we do have to be wary of that. when doing our analysis to see how these microbes could actually influence the isotope values. We have another great question about using isotopes for reconstructing precipitation. Rachel, I was wondering, I was wondering if you could comment. You did some awesome work looking at precipitation changes in Kenya. Would you be willing to answer that question?
Yeah, I mean, so hi everyone. Thanks for the talk. I reconstructed past precipitation changes, but of course not a direct sort of a measure of millimeters per rain, rather looking at amplitudes of variability through time. And it corresponds really nicely with orbital precession and other parameters. And so we do, there's good evidence showing that it is a good measure of past precipitation on those types of timescales. I did just see a paper about, by Badasso, on Ethiopian rainfall and how it correlates, or sorry, Ethiopian delta D and how it correlates with OLR. And that was a really interesting paper, and our interpretation of the amount effect is definitely something that we need to always be paying attention to and updating. Yeah, but I reconstruct past precipitation with delta D. That's what we use for these orbital timescales.
Thanks. Yeah, there have been quite a few studies looking in East Africa using delta D, like sourcing, like where the original source water is coming from, looking at lake records and the delta D from lake records, and looking at changes from Indian Ocean or Atlantic Ocean sourcing of water. So yeah, there's quite a bit of. I can put a list together if anyone's interested of readings for East Africa or just human evolution-based biomarker studies. I know I focused on East Africa, but there's work going on in South Africa. Alex is working in Armenia, right? So doing the European studies, so really cool. A few more questions. Can we use extracted organic residue from stone tools? This is something I've been wanting to do for like 3 or 4 years now, and I just constantly put it off. I know there is a lot of work going back a few years now, looking at lipids from pottery, and extracting leaf waxes and other types of fatty acids and stuff from pottery. I don't know if anyone has done it with stone tools yet. I know that it may be more recent material you'd be able to, if there was a way to show that what you're picking up is exactly from plant processing as opposed to something in the sediments. I guess you'd have to do a study of the tools itself, find the best way to extract it, maybe with an ultrasonicator, and then also compare the sediment all around the tools when they were excavated. One problem I see there is when doing the excavations, you better make sure you're on site to collect the samples so they're not contaminated, so they're not exposed on the surface. As archaeologists, we can be fairly dirty, and oftentimes we contaminate our own tools before we do any analysis, stone tools before we do any analysis. So yeah, so that's a great question. I've been waiting. I know somebody will probably get to it, but I've been trying to do stone tool lipid extractions unsuccessfully for a few years now, and I still have some samples sitting in the lab waiting to do that. Another question about sampling tools. I typically use like kitchen spoons or sampling spoons. They're sort of like the long rods. You can get them on Fisher Scientific. They're stainless steel. I always bring in the field typically isopropyl alcohol or some other type of organic solvent that can be cleaned with syringe filters in the field, or you can boil them ahead of time and keep them wrapped in oven cleaned combusted aluminum foil. But usually when I do my profile sampling, always stainless steel, even spoons. In some of the really tough sediments, I've even used screwdrivers, regular flathead screwdrivers that we clean with organic solvents ahead of time. You typically want to use glass or brown paper bags to collect samples. I know there are the plastic bags that can be used for, they're like the ultra clean plastic bags. I use those sometimes. when collecting some of this older material, but you just have to be careful that you're not contaminating your samples with any of the plastic material. I know for the leaf waxes, we don't typically have that issue, but if you're doing something like fecal biomarkers, you really have to be careful with the collection, the receptacles themselves. But yeah, so when I do my sampling, really a lot of it has to do with the integrity of the outcrop or the profile and how I can get really high resolution sampling that way. It'd be much easier if I had a corer, but sometimes, you know, just do what you have. So ultrasonication, negative impact biomarkers, not that I'm aware of. I've compared soxolate with ultrasonics before, and on the same sample, and I've typically had the same GC output and isotope ratios of the samples. I've never had an issue. The only thing with ultrasonication that I've always had problems with is that you can't use as much sample, so your yield isn't going to be as great when you do something like soxolate or accelerated or pressurized solvent extraction. Okay, some more on OLR, precipitation. Yeah. Any other questions? Anything else? I see that you're sharing papers. That's awesome. So there's some other. So I work in open air environments, but there are some other really cool human evolution studies looking at cave environments, and the questions they're trying to answer are whether humans were selecting specific plants on the landscape and then bringing those into the caves for food or for bedding or for burning, and then you can compare, you know, the alkanes with biomarkers for burning, for example. And I think that's really fascinating, using these cave records where plants wouldn't necessarily grow to answer questions about human selectivity. So there's some other really great research being done in caves as well, beyond some of the stuff that I talked about. And then I know we have a bunch of geochemists here, and they're doing the really pushing the boundaries of how these biomarkers can be used and coming up with new proxies for CO2 in the past, and how CO2 influences some proxies themselves. And I think that would be really great to introduce into archaeology at some point as well, as opposed to just looking at changes in precipitation and plant landscape. We can really incorporate some of these other factors. Okay. Anything else for right now? Like I said, if you want to email me questions later, or if you want the PowerPoint I have as a PDF I can send anybody, or I can share a reading list if you're interested in some of these other papers, or just general background readings on biomarkers. But really, that's what I have. Unless there are any other questions, I just like to thank you all for coming out and taking, you know, an hour out of your day to sit with us. And for those of you who were able to answer some questions, I really appreciate it. Yeah. Okay. Great. Thanks a lot. Good luck all. Hope everyone stays healthy and safe. And good luck getting back to real life whenever that happens. Thanks a lot.